Today's class will be on, we'll start the third unit now, that's cloning. Okay, before we move forward, so I welcome you all in today's class. It will be on gene cloning. So you can have an organism cloning also, like the dolly, that is this first sheep that was cloned, and then SKUAST also cloned on Noori by Dr. Riaz Shah, and that was a hit. So the animal cloning is different from gene cloning. Okay, there what we do, we take a cell, a somatic cell. Okay, an ovary cell, enucleate it, and then put a somatic cell inside the nucleus of a somatic cell. So that the organ that grows is the actual clone. So we're not dealing with that, we're dealing with gene cloning. So there are various aspects of gene cloning and there are various prerequisites of gene cloning. So what you require for gene cloning is, number one is your vector. Okay. Number two is your insert or gene that you want to clone. Okay. Three, you require a host. Okay. So 4, you require restriction enzymes. 5, you will require ligase. This is also an enzyme. So these are the prerequisites of cloning. So vector is a part of DNA that we use to transfer our gene of interest. Okay. In entomology, we call vector. A vector is kind of an insect or someone who transmits the disease, the virus, the bacteria from one plant to another. But in this case, the vector is a DNA, in this case, a plasmid DNA that will help in transferring our gene of interest, okay, into, or what we say, how to clone it, okay. So vector is nothing but a circular DNA, in this case, a plasmid DNA. Okay. Any circular DNA cannot act as a vector because there are some attributes of a vector that, and the prerequisites of a vector. If these prerequisites are not met by the DNA sequence, by the circular plasmid DNA sequence, we do not call it a vector. Okay. So the first and foremost property of a vector, okay, is that it should have an origin of replication. In this case, REC. Origin of replication is important entity of a vector. If a vector does not have this origin of replication, it will not be dispensable. What I mean by dispensability is that, for example, if you have a cell, bacterial cell, okay, and you have this vector inside, okay, if this vector does not have an origin of replication, in next mode of reproduction, say binary fission or whatever, 2 cells will be formed. Okay. If this does not have an origin of replication, this will not replicate. So one copy will stay as one copy. So this one copy will either go into this or this. Say, for example, it goes into this. So after one cycle, you have 2 cells and one cell has your vector. In the next cycle, this will divide again into 2 and this will again divide into 2. So you have now 4 cells, 4 daughter cells. And again, since this is not replicating, it does not have an origin of replication, it will only be here in one cell and one will be devoid of this vector. So after millions of, or say n cycles, you have millions of bacteria, but only one will have this plasmid, okay, or this vector. So we do not want that. We want that our vector should be dispensable, that because when you're cloning a gene, you need a lot of copies of the gene for future experimentation. You cannot have only one copy. So what we do, we put an origin of replication here so that this vector divides into two, okay, replicates, what we call as DNA replication, and one will go in one and another one will go in another one. So here now, this will also have, and here you will have. So after n number of cycles, if you have millions of bacteria produced from one bacterial cell, each and every cell will have a plasmid because of this origin of replication. So origin of replication is an important entity of a plasmid. It is an important attribute of a plasmid. If a plasmid does not have this origin of replication, this plasmid is not dispensable. So that means you'll lose it. Second thing that a vector should have is a marker. Okay, it's not this marker. Okay, it's marker for selection. In this case, an antibiotic resistance gene. For example, if this vector has an ampicillin gene, okay, the product of this gene, what it does, if this plasmid is inside a bacteria, okay, and this has an antibiotic resistance gene, in this case ampicillin. So what it does, this ampicillin gene or ampicillin resistance gene, it produces beta lactamase. This beta lactamase has a property that it breaks the beta lactam ring of ampicillin and renders it inactive. So now if somehow this plasmid goes into a cell, E. coli cell, okay, initially if this E. coli cell does not have this plasmid, if you grow this E. coli cell on a medium containing ampicillin, this cell will not be able to grow. So you will see no colonies because this bacteria is not resistant to ampicillin. However, if now you are taking this plasmid that has an ampicillin resistance gene, you put this in the bacteria, you put this in the bacterial cell or E. coli in this case. In lab, we use again that DH5 alpha strain. Okay, it's easier to maintain, so it's a modified strain. It's easier to work with. So now you put this vector inside this E. coli. Now you grow it on an ampicillin plate, LB medium that is ampicillin, or medium that has ampicillin antibiotic in it, and you grow this inside. You will be able to grow this because now this bacteria has Irshad, can you put your mic off? Everyone of you, just put your mics off. So, so now if this bacteria, this vector has an antibiotic resistant gene, and this vector is put inside an E. coli or a bacteria. Now we grow this E. coli on a medium that has ampicillin. This bacteria will be able to grow because now it has ampicillin resistance. Okay. So this is how we'll be able to know when we transform our E. coli, when we clone a gene, we transform our vector into the bacterial cells. Which bacteria harbors our plasmid will be known because they'll be able to grow. If the bacteria doesn't have a plasmid, it will not be able to grow. So this marker for selection is important, because once you take your gene, okay, you clone it, you put it in okay, you took a gene, you cloned it in the vector, okay. Now you put the vector inside the E. coli cell through a process called as transformation. Okay, transformation is a process through which a bacterial cell can take inside a foreign DNA. Okay, if one bacteria helps in putting a DNA fragment into another bacteria, that through a pilus, it's called as conjugation. Okay, and if a virus does the same, it's called as transduction. Okay, we do not want to go in detail. So simply, if we mix our cells with the DNA in presence of calcium chloride, okay, or DNA gets into the cell. The transformation practical will be doing. So, but you have millions of bacterial cells, you put millions of copies of this plasmid inside these, and there will be a chance that not all these bacterial cells will harbor a plasmid. Some will not accept a plasmid, okay? So if after transformation, you have just these two bacterial cells that have incorporated the plasmid, but these two have not. So when you grow this mixture on a plate, LB plate, and this plate has ampicillin in it, only this and this bacterial cell will be able to make colonies. Why? Because they have this vector, and the vector has ampicillin resistance gene. This and this colony, the vectors, E. coli cell will not be able to grow. They will not be able to make colonies because they do not have resistance against ampicillin. क्योंकि हमने media में we have put ampicillin antibiotic in the medium as a selection marker. Okay, so second important attribute of a vector is that it should have marker for selection. Third important thing that a vector should have is that it should have an MCS or a PCS, called as multiple cloning site or a polycloning site. This is the site that is rich in your restriction recognition sequences. Like if you go to the previous class or the previous YouTube video on restriction enzymes, we know that restriction enzymes are palindromic. We know that they are recognized by, they have a sequence, what we call the restriction site, and these are recognized by a specific restriction enzyme. So we know that multiple cloning sequence has a number of restriction sites. Okay, what we have to do, this restriction site, this multiple cloning site has restriction sites, like it has site for BAM-H1, okay, EcoR1, HIND3, okay, KPN1, and many more. Okay, there are like hundreds of restriction enzymes. So you have these many restriction sites here in the MCS or the polyclonal site. Because now what we have to do, this is our vector. Okay, we have to cut it at the multiple. This is the multiple cloning site here. Okay, we cut it, and this is our gene of interest. Then we ligate this gene of interest inside the vector. That's our goal. And put this vector inside a E. coli cell. Right? This is our goal. This is how we clone. So we look at the vector at the multiple cloning site. Okay, and the ends of the sequence of our gene of interest should also have this multiple cloning site. For example, I have made a vector for you. You see this DNA sequence. Okay, this is a DNA sequence. Okay, this is a plasmid DNA. So hope it's visible. This is a plasmid DNA. Okay, this is an open circular plasmid DNA. Okay, and if you see, I have kept some registration sites here. Okay, this is the multiple cloning site. Okay, this multiple cloning sites have 10, 15 recognition sequence of registration enzyme, different recognition enzymes. They are overlapping sequences so that in a smaller portion you have a lot of you have many registration sites. Okay, so for example, this site is GAATTC. GAATTC is the recognition sequence of anyone? Anyone? GAATTC is the recognition sequence of? Can you be a little bit louder? Can you be louder?
Equal.
Yes. Who is it?
Tamanna.
Oh, good. So it's the recognition sequence of eco R1. So what we have to do, this multiple cloning site ensures that हमारा जो vector है, हमारा जो gene of interest है, that goes inside the vector. Okay, it forms a plasmid. So what we do, so this is your restriction enzyme. Okay, these are called as molecular scissors. So, okay, are you able to see? Okay, so this will cut here. GAATTC, it will cut here, right? Then another cut on the same sequence because they are palindromic will be made here. Okay, so these sites have broken two phosphodiester bonds. Now you have these hydrogen bonds in between that is keeping them together. So what we do, we'll cut these. Okay, so this vector, circle vector, we have cut at a multiple cloning site. Okay, I'll keep it aside for a while. Now this is your gene of interest. This gene of interest we want to clone inside this vector. So the prerequisite is that you will use a restriction enzyme. You have 15, 20 restriction enzymes. So how will you select the restriction enzyme? So the restriction sequence that you will select should not be anywhere in this sequence. If your restriction site is between this sequence, because what you have to do when you design primers to amplify your gene, you put the same restriction site at the end of your primers so that when it amplifies in a PCR, you have a gene of interest flanking with your restriction site. For example, here you see I have kept GGA, okay, GAATTC again, okay. There also it was GAATTC. So you know that I have this gene of interest. Okay, I want to clone in the vector. Okay, so I'll use the multiple cloning site to open up the vector. I will cut it with the restriction enzyme here, EcoR1. I use EcoR1. I have to ensure that EcoR1 site is not anywhere between this gene. Because what we have to do, we have to also restrict this fragment with EcoR1. Why? Because I have kept EcoR1 at the flanking ends of this gene. Right? So you have a vector, okay, that I have cut with EcoR1. Okay, sorry, a vector that I have cut, and the insert that I have also cut with EcoR1. Now the ends are compatible. If you cut one EcoR1 DNA here and another EcoR1 DNA, you cut another DNA in say United States with EcoR1, they'll have again the compatible ends because they recognize the same sequence. In this case, if this is your insert, okay, now you have on one end you have again GAATTC. Can you see? Again you have GAATTC right here. Again, okay, because we are digesting the insert also with the restriction enzyme that I used to digest the vector, so that same restriction sites are at the end of both because they are compatible. So we'll cut it like this. Okay, so this is the end you got. These are called as compatible ends, and the ends they have produced are sticky ends. So GAATTC again. Here. Is it visible? Okay. Now you see you have an insert that has compatible ends with your vector. How? Now this is your insert. I'll keep it like this. Okay. This is your vector, right? When you mix the two and add ligase enzyme, what will happen? This will form A will pair with A will pair with A, T will pair with A, T will pair with A. Sorry, A will pair with T and T will pair with A. So they'll form these hydrogen bonds. And then with the ligase will connect this the phosphodiester bond and this phosphodiester bond. So this is your one end. This is your another end. Okay. So you see here. You again have right. So in this way, now you have a vector that has your insert. And how did we do that? We restricted it with a restriction enzyme that was in the multiple coding site. So multiple coding site is a site that is rich in your restriction recognition sequences, and it is a site where we put our gene of interest. Fourth property is that your vector should be small in size. Okay, otherwise it will be just trying like to get an elephant inside the house. Okay, so that's why we use the quality of a good vector is that it should have small size. It's one of the attributes. So that's why the multiple cloning site here is overlapping. That means in a smaller region, you have more number of multiple cloning. If you Google multiple cloning sites of a vector, you will see that if this is the recognition sequence of equarvan, recognition sequence of say biometric will be like this. So they'll have an overlapping region so that you can make the vector size smaller. Smaller vector, better to control and better to clone things in it. So another thing is that the vector should be small in size. Then you have another property of a vector is that it should have a marker for recombination. This marker for recombination is here. This marker for recombination lies, okay, I can say multiple cloning site lies in the marker for recombination. Why do we require marker for recombination is that, for example, you took a mixture of plasmids, okay? So this is a mixture of plasmids, okay? Right? And some have this insert. You took a, okay, see what happens. You took a plasmid, okay? You restrict and digest it, okay? You mix it with an insert that has the same sequences at the end. You ligated it to, and now you have a sequence like this, right? But the thing is that if you see a vector, okay, this is your insert. Somehow the insert does not get incorporated, okay, does not get cloned. So, but when you have restricted a vector with a restriction enzyme, you see the sequences that they produce, okay, they are compatible ends. They can again religate or reanneal, right? So chances are that when you put them in a solution of ligase, okay, ligase will bind them together back, right? There's a chance, okay, because the ends are compatible. It's a vector, okay? Or either an insert can go in between. Both things can happen. So that's why we require marker for recombination. Because for example, say these are the vectors, okay, and say in two of the vectors you have your insert or gene of interest, but others religated. They are empty vectors. When you transform these these vectors in the E. coli, okay, and then you plate those E. coli on an LB plate, all of these will be able to grow because you have ampicillin here, right? And all of these vectors have ampicillin. So you have vector, 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 vector. Now you do not know that which of these have your gene of interest, right? Because here you have only two of these vectors have gene of interest, but you do not know because they will also grow, these will also grow. So you are confused. So you have a mixture where you have empty vector and then you have gene of interest. Okay. So to overcome this problem, we use a marker for recombination. So marker for recombination, for example, in this case, now we use LAG-Z. LAG-Z is a gene that breaks X-Gol. Okay. X-Gol is a compound, okay, or a molecule. When X-Gol breaks into two molecules, one of them is blue in color. It is a blue color. Other one is colorless, right? So if we see now the situation, you have a multiple cloning site. Let's draw it separately. So you have, this is your multiple cloning site. This is your marker, right? And you have another condition also. You have marker and you have this MCS. This way, this LAG-Z, this is LAG-Z, right? LAG-Z marker. If we have not digested this with restriction enzyme, this LAG-Z is functional. Okay. And if we take a restriction enzyme, we break this LAG-Z because multiple clone site is in between the LAG-Z. But when we ligate it, it again ligates back. That means we are rejoining the gene again back. So that means it will again be functional. So if a vector is there, you restriction digest it because, and then ligate again it will be functional because you are undoing the function. You are ligating it back. So if the vector is empty, no insert has been incorporated between, LAG-Z will be functional. And if an insert comes in between, this is an insert, okay, it gets cloned between your gene. So now your lag Z is from here to here and from here to here, right? हमने इसको तोड़ दिया, इसके बीच में हमने gene of interest डाल दिया. So now we have two scenarios, okay? One is that we restriction digested it, okay? And then it got ligated back. So this will produce functional lag Z. Good. In this case, when we restriction digested it, we cloned our gene of interest between that, right? Because we are adding or insert. These are multiple cloning sites on both sides. And this is your lag Z part 1 and lag Z part 2. This will not be functional because now you have an insert in between. That's a big gene. So it will make this gene dysfunctional. So lag Z is not functional here. Now there are two circumstances. If this goes in the E. coli cell, if this goes in the E. coli cell. So this time what we do, we plate them on ampicillin, right? Because this is a part of a vector that has ampicillin gene here. This is part of a vector that has ampicillin gene here. Okay, so we grow it in ampicillin, both will grow, right? But here what we do, we add X-gall, a chromogen, like ampicillin in the medium. Now both these cells will grow, right? And they'll form a colony because all are resistant to your ampicillin. But now here what will happen? This colony or this vector, which does not have an insert, its lag Z is functional. It will produce lag Z, functional lag Z, and that will degrade the X-gall in your medium. Once the X-gall is degraded, it will give blue color because one part of that, what we call the product of X-gall, it breaks into two parts, as I told. Okay, 1 and 2. The one part gives a blue color. So these colonies will appear blue because of a functional lag Z. But blue colonies are not our colonies of interest because we know that they don't have a gene of interest. If they would have a gene of interest, their lag Z will not be functional. When lag Z is not functional, they will not be able to degrade X-gall. When X-gall is not degraded, they will use simple off-white colonies. So that's why we require marker for recombination or marker for screening. This marker helps us to differentiate between those vectors or those vectors that are empty, that do not have a gene of interest, we do not require them, and those vectors that have our gene of interest, right? Good. Another important property of a cloning vector is that it should have high copy number.